Hello, I'm Xiaoyue from the Duran Lab at Carnegie Mellon University. My talk today is on simulating the evolution of protein domain architectures. Simulation is an essential technique for studying evolution, where most processes of interest act on the timescales that exceed a human lifetime. Simulating protein family evolution is an active area of research that has produced a wealth of sophisticated models and software. However, most work has not considered the special challenges of simulating multi-domain proteins like this one. Domain order and co-occurrence in these proteins is highly constrained. Today, I'm going to tell you about our model of domain architecture evolution and how we use it to simulate evolutionary trajectories that reflect the constraints on domain order and co-occurrence observed in nature. The overarching goal of this work is to simulate protein domain architecture evolution in a way that recapitulates the properties of genuine domain architectures. I'm going to start by introducing multi-domain proteins and their properties. Then, I will introduce some existing protein evolution simulators and discuss why simulating domain architecture evolution is not a plug-and-play problem. Next, I'm going to introduce our model of domain architecture evolution and empirical evaluation of our simulator. Multi-domain proteins are mosaics of sequence segments that encode structural or functional units called domains. These segments are found in otherwise unrelated proteins, and they can fold independently in many different contexts. For example, the Sark protein contains 3 domains, the SH3, SH2, and the kinase domains, which appear here as separate folds. This is a cartoon representation of the sequence that encodes the Sark protein. These colored icons show the locations of the segments that encode the SH3, SH2, and kinase domains respectively. The domain content of a protein can be determined by scanning its amino acid sequence with probabilistic domain models from the domain database, such as PFAM or superfamily. This is an example of a domain HMM, shown as a logo that represents the amino acid composition of each site. Scanning the sequence with these HMMs identifies the starting and ending position of each domain present in the sequence. The domain architecture of a protein is an abstract representation consisting of the sequence of domains in a protein in N to C terminal order. The domain architecture representation is a substantial simplification. It ignores the specific amino acid sequence of the domains and the linker sequence connecting them. Despite this simplification, the DA representation has proved extremely useful for studying protein function and evolution, because it reveals the large-scale organization of protein architecture and modularity. Shown in this box is just a sample of types of studies that have used this representation. Multi-domain proteins evolve through a variety of molecular mechanisms that can change the domain content of the protein. In a domain architecture representation, we can treat these mechanisms as 2 abstract events. A domain architecture can gain a domain through insertion or internal duplication, or lose a domain via deletion. In principle, these events have the potential to generate any combination of domains over the course of evolution. In practice, it appears that the set of successful changes to domain architectures is extremely constrained. Only a tiny fraction of possible domain combinations are observed in nature. For example, in primate proteomes, we only observe 0.4% of all possible domain pairs and 0.0006% of all possible domain triples. We used a randomization test to see whether this is surprising. The randomization process permutes the assignment of domains to domain architectures, but it preserves the number of domain architectures, the number of domains in each domain architecture, and the number of instances of each domain. As a result, Genuine DAs have much fewer domain combinations than expected, given the number of domains. Another good example is that zinc finger immunoglobulin are the most common primate domains, but never co-occur in the same protein. But they co-occur 344 times on average in randomized DAs. As we have just seen, domain adjacency and co-occurrence are stringently constrained. A realistic simulator of domain architecture evolution should reproduce these constraints. I'm going to review some existing protein evolution simulators and discuss why we need more to simulate domain architecture evolution. If we ignore, for a moment, the problem of modeling gain and loss of protein modules, proteins evolve on two levels of biological organization: gene events and sequence events. Gene duplication, transfer, and loss are events that expand and contract the size of a protein family. These are typically modeled by a birth-death process on a species tree, resulting in a gene tree. The evolution of the sequence is then modeled by instantiating a sequence at the root of the gene tree and simulating the evolution of that sequence over the branches of the tree. Both of these problems are well studied, and many good software tools are available. Simulating multi-domain proteins requires modeling three levels of biological organization: gene events, domain events, and sequence events. Gene events are simulated as before, resulting in a gene tree. Next, domain events are simulated on the resulting gene tree, again using a birth-death gain model. However, this process is carried out several times, once for each constituent domain. The result is a mosaic of sequence segments and a tree for each segment. Sequence evolution models are applied as before. However, the evolution of each sequence segment must be simulated separately because different domains may be evolving according to different models and different rates. SAGE-5, which is based on gene follow data, simulates modular protein families using this three-level approach. EvolCompGen is specifically designed to support different evolutionary models in different regions of the sequence. The three-level approach I have just described supports simulation of modular protein evolution, where the modules are arbitrary sequence fragments. In order to model the evolution of multi-domain families composed of genuine domain families, some additional machinery is needed. The sequence evolution simulator must reflect the site-specific constraints that preserve the characteristic sequence features of the domain, such as this conserved tryptophan in the SH3 domain. The Revolver program from Ingo Ebersberger's lab does this using a sequence evolution model that is guided by a domain family HMM. Similarly, in order to simulate realistic domain architectures, the domain event simulator must reflect the constraints on domain co-occurrence seen in nature. The design of a domain architecture simulator that achieves this is the subject of the work I'm presenting to you today. Now I'm going to tell you about our model of domain architecture evolution. We present a domain architecture evolution simulator specifically designed to generate domain architectures that mimic the constraints on domain order and co-occurrence observed in nature. The simulator is a Markov model in which each state corresponds to a domain architecture, where the domains are sampled from a domain alphabet specified by the user. Typically, the alphabet will be the set of all domains encoded in one or more proteomes provided as training data. The simulator repeatedly proposes a new state and then accepts or rejects the proposed state. We need a function that proposes new states and a basis for accepting or rejecting the proposed state in such a way that the simulator visits states that correspond to domain architectures with properties that are typical of genuine domain architectures. Transitions to adjacent states correspond to gaining or losing a single domain. For example, given a domain architecture of length 3, there are 3 states that correspond to loss events. The set of adjacent gain states is much larger. There are 4 sites in the domain architecture where a gain can occur. For each of these sites, any domain in the domain alphabet is a candidate for insertion. To simulate evolution of domain architectures, we adapt the Metropolis-Hastings algorithm. At each time step, a new state is proposed by selecting an adjacent state uniformly at random. The probability of acceptance is determined by the ratio of the probabilities of the new domain architecture and the current domain architecture. Of course, we don't know the probability of a domain architecture, and we can only estimate it. And importantly, we want our estimate of these probabilities to reflect the constraints of domain order and adjacency. In other words, adjacent states that obey the constraints better should have a higher probability of acceptance. In the current implementation of our simulator, we use a first-order approximation where the probability of a domain architecture is the product of conditional probabilities. The conditional probabilities are estimated from bigram frequencies observed in genuine data. This model has been implemented in a Python package, the Markov. The user specifies chain length, that is the number of new state proposals, T, and provides a training data set, from which simulated parameters are estimated. The initial state is a single domain architecture, where the domain is selected according to its frequency in the training data. At each time step, a new state is proposed by inserting or deleting a domain from the current domain architecture. The proposed state is accepted based on estimated state probabilities that reflect the constraints of domain co-occurrence observed in the training data. Next, I'm going to show empirical results of our simulator. Recall that our goal is to simulate domain architectures that recapitulate properties of genuine domain architectures. Have we realized our goal? How well does the model recapitulate biology? We tested the simulator on 4 metazoan datasets. Since multi-domain architectures in metazoa tend to be longer and more varied than in other branches of the tree of life. In each case, we generated a set of simulated domain architectures equal in number to the set of unique domain architectures in the training data, and compared the properties of the 2 sets. For our empirical evaluation, we downloaded domain annotations from the superfamily database. We extract bigram frequencies. These are used for a first-order approximation. We add pseudo-counts for unseen bigrams. Domain combination statistics vary across the metazoa. To assess the impact of this effect, we experimented with training data from metazoan lineages. 2 vertebrate lineages, 1 invertebrate bilaterian lineage, and 1 early branching lineage that diverged prior to the base of the bilateria. The properties that we compare include domain architecture lengths, domain combinations, domain promiscuity, tandem repeats, and event positions. We first consider the distribution of domain architecture lengths as a function of chain length. We ran chain lengths ranging from 7.5 thousand to 12.8 million time steps. Here I'm showing you final domain architecture length distributions for various chain lengths from the primate simulations. And this is the genuine length distribution on the right. The genuine distribution contains some outliers with very long domain architectures. These huge outliers are orthologs of titin, a muscle protein that can contain as many as 253 domains. Our simulator did not generate a domain architecture of this magnitude in any of our experiments. However, if we leave these outliers and zoom in, you can see as we increase the chain length, the mean domain architecture length converges to the mean in the genuine data. This demonstrates the stability of our model. It's able to achieve a balance between domain gain and loss that produces realistic domain architecture lengths. We see similar behavior in the other 3 datasets. Like primates, fish have a small number of very long domain architectures that substantially exceed the lengths of the longest simulated domain architectures. These outliers are not seen in the two invertebrate datasets. We also see faster convergence to the mean domain architecture length in the invertebrate datasets. That is, a shorter chain length is sufficient to obtain domain architecture lengths with the same mean as the genuine data. A chain length of 3.2 million time steps was sufficient for all 4 datasets, so we used this chain length in all subsequent experiments. Next, as a sanity check, we compared bigram and trigram frequencies in the genuine and simulated data. Since bigram frequencies were used to estimate the transition probabilities, disagreement between the simulated and genuine frequencies would be a red flag. As expected, simulated and genuine bigram frequencies are in very strong agreement, with a Pearson correlation coefficient of 0.997. For comparison, we calculated bigram frequencies for randomized domain architectures. The Pearson correlation coefficient, averaged over 1,000 iterates, was 0.314, shown as a cross-hatching on this bar chart. For visualization purposes, we selected one of the 1,000 iterates for inclusion in a scatter plot on the left. We see similar behavior for both bigram and trigram frequencies in all 4 datasets, with very strong agreements between the simulated and genuine domain architectures, but not between randomized and genuine architectures. We next considered properties of domains and domain architectures that are not directly related to bigram frequencies. Protein domains vary in their propensity to co-occur with other domains, a property that is sometimes called domain promiscuity. For example, the SH2 domain is highly promiscuous. It co-occurs with many different domains in varied domain architectures. In contrast, the SNF-like domain is not promiscuous. It only occurs by itself. To determine whether domains in our simulated domain architectures recapitulate their genuine promiscuity levels, we quantified promiscuity using 2 measures from the literature. Because of time constraints, I won't give the equations. Yuting will discuss these and other measures in the talk immediately after this one. These calculations are also described in our Proceedings paper. As you see, domain promiscuity is preserved in simulated domain architectures. Some domains are frequently found in repeated arrays, others are not. The Markov does not distinguish between gains by duplication and gains by insertion. Since the Markov does not model duplications explicitly, we asked whether domains that form tandem repeats in genuine domain architectures also participate in tandem repeats in simulated domain architectures. We first compared the mean length of tandem arrays in simulated and genuine domain architectures. Surprisingly, we see strong agreement, especially in the fish and invertebrate datasets. The agreement is slightly weaker in the primate dataset. Recall that primates have a few very long multi-domain proteins, such as Titan, that contain tandem repeats. The Markov's longest simulated domain architectures fell short in comparison. We also considered the most repetitive domains in each dataset and found that the 10 domains with the most repeat copies were the same in both datasets, although the ranking is slightly different. This demonstrates that the Markov can model tandem repeats despite the lack of an explicit duplication function. Several studies using a variety of comparative approaches report evidence that evolutionary events occur more commonly at the termini of multi-domain proteins than in the interior. This may be due to mutational processes result in modifications at the N or C terminal of a protein, including gene fusion, acquisition of premature stop codons, and acquisition of an alternative transcriptional or translational start signal. Proteins may also be more resilient to changes at the termini, since those regions are often located on the surface of the folded protein, rather than contributing to the structural integrity of the protein core. To determine whether the simulator recapitulates this behavior, We tabulated the positions of accepted gain and loss events in simulated domain architectures of lengths 3 to 8 in the primary simulations. Remarkably, there is a clear preference for terminal over internal gains and losses. To summarize, there are many challenges in simulating domain architectures. The Markov is our proposed solution. It uses Metropolis Hastings strategy, uses data-driven transition probabilities estimated from genuine bigram frequencies, and supports species-specific domain architecture simulation. Our empirical evaluation shows that simulated domain architectures recapitulate properties of genuine DAs. We are currently working on simulation on a tree in other lineages, distinguishing insertions and internal duplications, multi-domain events, and other approaches to estimating state probabilities. Ultimately, we want to combine domain architecture simulation with simulation of other aspects of protein evolution to obtain a comprehensive multi-domain simulator. I would like to thank all my colleagues in the Duran Lab, thank Lars, Ingo, and Arn for helpful discussions, and thank you for your attention.